So the electron transport chain, this last step in cell respiration, the reason it's called a chain, it's actually a chain of proteins. And these proteins are in the inner mitochondrial membrane called the cristae. So a quick reminder, this is a mitochondria, and this is the cristae, this inner membrane. So when they're showing this set of proteins, these are the little molecules that are in the electron transport chain. They're just sort of lined up in the in the membrane. And you don't have to know the names of any of these specific molecules. What's going to happen at this point is all the NADH and the FADH2 are going to pass their electrons down this electron transport chain. So they are going to go back to being their oxidized forms. This will go back to being NAD8+. This will go back to being FAD. And the hydrogens are going to break off. Now, the next slide is going to clarify this, but I just want to make a quick comment. Only electrons go down the electron transport chain, not hydrogens. So when when the H breaks off, the H actually breaks into two things. A hydrogen ion, which is technically a proton, because hydrogen is number 1 on the periodic table. It has 1 proton and 1 electron. And an electron. So that H from NADH and the same with the H from FAD, it's going to split into a hydrogen ion and an electron. The electron is what goes down this electron transport chain. Just the electron, not the hydrogen ion. What's going to happen to the hydrogen ions, and you'll see this on the next slide, is that they're going to get pumped through these membranes and they're going to accumulate in this space between the outer and inner mitochondrial membrane. So this is going to come up on the next slide, but I wanted to clarify that. Remember that as the electrons flow down the chain, this actually releases energy. And that energy is what's actually going to drive the pumping of the hydrogens into the space that I'll show you in just a second. The other thing is, and this I left off, I realized later, of your outline, but this is really important. This is where the oxygen becomes necessary. Oxygen is what's pulling the electrons down the electron transport chain. Oxygen is highly electronegative. So it's sort of sucking the electrons towards it, pulling them down the chain. And at the end of the electron transport chain, what's going to happen is 2 hydrogens are going to combine with oxygen and the electrons, and it's going to form water. So this is the only step in cell respiration where we finally see what happens to the oxygen. We didn't use oxygen for glycolysis. We didn't use any oxygen in the Krebs cycle, even though the Krebs cycle won't happen if oxygen isn't present. We didn't technically use oxygen. This is where the oxygen comes in. Oxygen is the final electron acceptor at the end of the electron transport chain. The other thing to note here is that this does not actually make the ATP. In other words, the electron transport chain does not directly generate the ATP. They are going to be made at the end of the electron transport chain as a final step, but the actual moving of electrons down the electron transport chain doesn't directly make the ATP. So all we've said so far is that NADH and FADH2 lose their hydrogens at this point. The electrons are going to go down an electron transport chain, or what we call the electron transport chain. And if we go to our next slide here, the hydrogen ions, which are actually now just a proton, they are not traveling down the electron transport chain, but they are getting pumped into what we call the intermembrane space, which is the gap between the inner membrane of the mitochondrion and the outer membrane of the mitochondrion. So the hydrogen ions are building up here in this space. The electrons from NAD and FAD are traveling down the electron transport chain. So this is our electrons moving down the electron transport chain, but the hydrogens are getting pumped into this intermembrane space. And they basically build up in that space and then re-enter, come back through ATP synthase, the enzyme. And that is what actually makes the ATP. The most direct source of making the ATP is the hydrogen ions, the protons, moving through ATP synthase. That's what generates the ATP. And that is technically what we would call a proton gradient, a difference in charge on the two sides of the membrane due to the buildup of the hydrogen ions in the intermembrane space. And I'm going to show you an animation of this that I think will completely clarify it, because to me, it's complicated. But I think when you see the animation, it'll clarify it for you. Also, for every NADH, and this is where the numbers do come in, for every NADH that enters the electron transport chain, 3 ATP can be generated. And for every FADH2 that enters the electron transport chain, 2 ATP can be generated. Some people find that confusing because they think, oh, but shouldn't it generate more because it has more hydrogens? But it doesn't have to do with how many hydrogens it has. It has to do with where the electrons enter the chain, and it turns out the ones from FADH enter the electron transport chain a little further down the pathway. So that's, you don't have to know that reason, but I know that some people get confused because they're like, well, if it has more hydrogens, shouldn't it make more? And it doesn't. So NADH makes 3 ATP. Every FADH generates 2 ATP. So this is where all your ATP are going to end up being made. You've only made 4 prior to this step. And remember, this is called chemiosmotic or oxidative phosphorylation because it's relying on this gradient that's established. I have a couple of pictures. So this one is showing the Krebs cycle happening in the matrix of the mitochondria. The CO2 leaves, that's our waste product. And then they're showing here, it's not a great drawing, but it's showing the NADH is here and the FADH2. And they're showing the electrons going down this electron transport chain, the hydrogens getting pumped into this intermembrane space. These next 2 diagrams also show it. Here's the first step. The hydrogen's breaking off of NAD, and the electron is the yellow traveling down the electron transport chain. The hydrogen's building up here. And then in our last step, this big buildup of hydrogen ions, they flow through ATP synthase, and that's what generates all of the ATP. So let me show you a little summary. Glycolysis, you make a net of 2 ATP. Citric acid cycle made 2 ATP. And then here in the electron transport chain, it makes an additional 32 to 34 ATP. So our total from cell respiration ends up being somewhere in the neighborhood of 36 to 38. So here's a summary. So glycolysis made a net of 2, we just said. The citric acid cycle made 2 more. Keep in mind, and I added this here at the bottom just to clarify, that the ones that were made in glycolysis and in the citric acid cycle were made by that other way of making ATP, substrate level phosphorylation, which is the way of making ATP sort of one at a time that's not very efficient. And then in the electron transport chain, all the NADH and FADH2 get rid of their hydrogens and their electrons, and ATP synthase then generates an additional 32. Depending on the book you look at, you may see 32 or you may see 34. On a test, it's not important that you memorize that total number of being either 36 or 38 specifically. If I was going to ask you how many total ATP are made in cell respiration, I wouldn't have both 36 and 38 as choices. I would have really spread out stuff like 36, 45, 20, and 7. In which case you know that the one that's either 36 or 38 is going to be your answer. So don't worry about memorizing, why do some books say 36 and some books say 38? For the total ATP, some will say 32 here, some will say 34 here. It has to do with the coenzymes, but it's not important to memorize which number is the correct number. Just know it's around 32 to 34, and that the total ATP made are 36 to 38. All right, let me show you the video, this little video clip, and then we'll come back to this. This sort of summarizes everything. Let me get right to here. That, and this should help with clarifying it. Here.
The electron transport chain is an array of molecules, mostly proteins, built into the inner membrane of the mitochondria. NADH gives up its high-energy electrons to the first complex in the electron transport chain. The electrons move from one member of the chain to the next, giving up their energy as they are pulled from NADH toward highly electronegative oxygen. The energy given up by the flow of electrons is used to pump hydrogen ions from the mitochondrial matrix into the intermembrane space. Oxygen captures the electrons in the very last step in electron transport. The last complex adds a pair of electrons to an oxygen atom and 2 hydrogen ions, forming water. The The electron transport chain has used the energy of moving electrons to pump hydrogen ions into the intermembrane space. This buildup of hydrogen ions, like water behind a dam, stores the potential energy that was originally in the bonds of glucose molecules. The backed up hydrogen ions give up their energy when they diffuse through a special protein in the membrane called ATP synthase. As hydrogen ions flow down their concentration gradient, ATP synthase captures their energy to make ATP. This mode of ATP production is called oxidative phosphorylation because it is powered by the transfer of electrons to oxygen. Under normal conditions, almost all the ATP produced in the process of cellular respiration is manufactured by electron transport and oxidative phosphorylation, about 34 ATPs for every glucose consumed.
This illustration summarizes cellular respiration. Glycolysis and the citric acid cycle produce a small amount of ATP via substrate-level phosphorylation, but most of the cell's ATP is made via oxidative phosphorylation, when NADH and FADH2 produced in glycolysis and the citric acid cycle give up to oxygen the electrons obtained from organic molecules.
Okay, alright, so this sort of summarizes the pathway that the energy flows. The energy starts in glucose. Remember, glucose got split into 2 pyruvates, so technically each pyruvate has less potential energy in it than the original glucose did. A little bit of energy was trapped in NADH, that's why I included that there. The energy from pyruvate gets passed to another molecule called acetyl-CoA. You don't have to know that molecule, but I included it because it is a step. Acetyl-CoA is technically what feeds into the Krebs cycle. It's the starter molecule that enters the Krebs cycle. By the end of the Krebs cycle, the energy is now trapped in NADH and FADH2, a bunch of them, not in just one of them. And then those go down the electron transport chain, and our final goal is to trap our energy in ATP. So if you were putting it in order from what has the most energy to what has the least energy, glucose has the most energy. And we break the glucose into several ATP. Each ATP has less energy than anything else here. Which of these two has more energy, NADH or FADH2? NADH, why? Because in the electron transport chain, every NADH has enough potential energy to make 3 ATP, and FADH2 only has enough potential energy to make 2 ATP. And then in the end, like I said, ATP technically has less energy in it than NADH, because you are making 3 ATPs for every NADH. So that's sort of the pathway that the energy flows as far as during cell respiration. And the last slide here is just, in a perfect world, there would be 36, or, again, some books will say 38. It has to do with these NADHs that were made in glycolysis. Technically, NADH that's made in the cytoplasm can't pass directly into the mitochondria. So what actually happens in most organisms is that these NADHs pass their electrons to FAD, and since FAD only makes 2 ATP each, you end up making a little bit less. But you don't have to understand that. But that is why there's a discrepancy. So some books will say 36, some books will say 38. That specific number, it doesn't matter which one you know. It's fine either way. And again, a majority of them were made in the electron transport chain. And I say in a perfect world because it's sort of like your car. You know, there's a sticker when you buy your car saying how many miles per gallon your car is going to get. It's probably not really going to get that many miles, especially if you're driving in the city, blah, blah, blah. Same is true in our mitochondria. That's the perfect situation. That's the maximum. But you never know. All kinds of variables, temperature and all kinds of things going on in the cell may make it a little bit less than that. The last slide here is just to show you, just a reminder that even though we learned sugar going through glucose, going through glycolysis, technically you can use other things for energy. You can use proteins. They'll just get converted to amino acids. Those will get broken down and fed into the Krebs cycle and into the electron transport chain. And you can break down fats. Fats are made of fatty acid and glycerol. Glycerol will go right into glycolysis in one of the steps, and the fatty acids will enter right here as acetyl-CoA that enters the citric acid cycle. So you can use other sources to make ATP, But glucose is the main one that your body's using. That's what's floating in your blood right now that, that your cells are using mainly, and that's why we